about different things a bit. But to, so we just learned something about rate of adaptation, right? Or it could be some complicated thing, okay? So we'll do some bunch of exercises, and also I want to introduce some new things quickly, hopefully. So tell me the following thing. So somebody mentioned molecular clock. Molecular clock, right? So we just learned that the rate of adaptation is equal to 1 by NUB 2SB into SB, right? Right? So this was for a beneficial mutation. I would like you to think about this and tell me, what if I have a mutation which is neutral? So now fitness is not changing with time, right? Because it's a neutral mutation. What's the rate at which neutral mutations accumulate? This is beneficial. So all you have to do is to use this kind of idea. Again, feel free to chat, discuss, but just avoid Googling. You come. I have answered lots of your questions, so I think it's time you answered mine. Come to the board. Let's give it a try. Come on. Come. Come. Arif. Come on, help. Come. So those who didn't come to the board yesterday, today is your day. Try something. Huh? Okay, want to think again? Okay. Any of the volunteers? Or you haven't come to the board? Did you come to the board? Oh. No, but first row I'm not asking. So you're safe. Oh yeah, right. Hey guys, come please. There's so many nakras and all. Come. Yeah, the fourth person looking away. Come. Just try it. Come on. I made so much effort. So I expect something from you. You're coming? Oh, good. Come. Bach gaye. Come. So, Pratik, I'm going to ask you yesterday's question. Selection, mutation, drift. How does the distribution look like? Better get started. Come. Can we have a mic, please? Yeah. Do you want some mic? Explain. Explain.
So in case of when it's neutral mutation, selection won't act. So we will only consider mutation and drift. So this takes into account the drift factor and 2SB because we calculated in the previous one.
The fixation probability of what? What kind of mutation?
Beneficial. So we can just write this. Yes.
So what is Sn?
So neutral N for
what
neutral.
Is Sn? Selection coefficient N is 0, na?
Okay.
So R is 0. Neutral mutations don't accumulate.
But they do.
They do.
Yes.
So very good. That's progress. So something is wrong.
Yes.
What is wrong? You're almost there. Denominator is correct. Numerator. So you're right. This should be the fixation probability of
neutral mutation.
Neutral mutation. What's the fixation probability of neutral mutation? 1 by n. Right? So what did you get?
1 by n by 1 by n.
So simplify.
So n un by n.
Yeah, go ahead. Done?
Yes.
Okay, correct, very nice. People agree? Yeah, good. Thank you. Right? So that's what we are talking about, right? Molecular clock. So this is the same calculation. All you have to do is to change things a little bit. So if you understood any of them, second one you can do. Mutatis mutandis, how do you say that? That means making appropriate changes. Right? Is this okay? Yeah. Okay. Yesterday's question. Selection, mutation, drift. How does distribution look like? Selection, mutation, and drift. Probability distribution without selection was? What was it yesterday? Yeah, it was like this, U-shaped and like this, right? And I asked you guys to think what happens if you put in selection as well. So how does it look? How many people tried? Very good. Who else? The usual suspects. Anybody unusual? Third person. You came yesterday. Okay. Who wants to come? Okay, so that's a little bit more involved, so I'll come to that a little later. Let me talk. Okay. Okay, so it's fine. I mean, let me just go ahead a little bit because I wanted to introduce two things because time is very less what to do. See, yesterday I talked a lot about population genetics, and today just before 15 minutes, again some population genetics, right? And we said there are these, evolutionary forces, we put them together and do with it. And it describes things at the genetic level. That's one approach, okay? Do you know of other approaches to understand evolution? Any other approach? Or other approaches? I'm not asking for alternative approaches, I'm asking for complementary approaches. Do you know of any other ones? So this is, I would call, classical population genetics. Is that what the whole world is doing? Who is interested in evolution? Any other theories? What else is there? Huh? Let's stick with single species. So phylogenetics I'm not considering because I'm going to think only of single, and that's what population genetics, most of it does. Yeah. Anything else? Collison theory, which is also a population genetics theory, but the idea is the following. In this PopGen, I'll explain to you with an example. If I give you some properties, the population size, mutation rate, selection coefficient, and some bunch of rules, like I did yesterday for Wright-Fisher process, I'm asking, given today's, what happens forward in time? As time goes on, what would I see to the allele frequencies? Collison theory, it started in 1980s. I think there's some history to it, but there it's a backward time approach. This is also PopGen, but it's a backward time. So the question that one is asking here is that if I give you some samples today, just by looking at today's sample, can I say whatever diversity or some properties that I'm seeing, how were they shaped? What happened in the past? By just looking at today's sample, can I say something? Looks like science fiction, no? Sounds like science fiction. But it's not like that. I mean, there's a very nice, very beautiful maths, and it's a lot of its contribution has to come from mathematicians. And the name of one person which is associated with is Professor Kingman. Okay? So I'll explain a little bit of that. It's called Kingman's Collison, but there are other versions also. So I'll explain this. Okay. Anything else? Are there other approaches to understand evolution? That's it. It's kind of boring, no? I mean,
character reconstruction and fluorescent.
Sorry?
Character reconstruction and fluorescent.
I don't know, what is that?
Basically, you have one character which is you're just using Markovian processes to go back in time.
Okay. No, so I don't know what that is, but I don't know. I don't know at all. So maybe there's another one which I don't know. Anything else that you can say? You heard about game theory. Didn't you? Right? Evolutionary dynamics is in game theory. You heard this already this week. Okay, so this I don't know enough, so I won't say. And there's one more called adaptive dynamics. Again, I don't know enough to discuss this. But I left some space here for one more theory. What is it that I have not spoken about? So we're using all genetics, allele frequencies, this mutation, that mutation, BRCA1 or whatever. But what is that selection acts on? Phenotype. So did I speak about phenotype at all? When did I speak about phenotype? Did I? I just said genetic, like this locus, this sequence, hoo-ha. So what is that theory that deals with phenotype? Okay. That's called quantitative genetics. You heard about quantitative traits, no? Okay. So this one, the classical traditional one, deals only with phenotypes. You don't know much about what's happening at genetic level, so you just make some reasonable assumptions and work only at the phenotypic level. You don't not care, sometimes you do, but you don't know much about them, so you just work at the phenotypic level. And then there is population genetics, coalescent or classical one, that's at the genetic level. Is this a good state of affairs? You do your job, I'll do my job. What shall we do? Merge? Merge, I don't know. Any better suggestion? Not merge. Huh? No. This is, I'm saying this level. Yeah. So this is one level, this is another level. Yeah. Connect them, right? So that means, so why don't we just connect those two levels? What do I need to connect the phenotypic evolution and genotypic evolution? What do I need? How would I go about doing it, let's say in principle? Yeah, very good. So we need a phenotype-genotype map. That means if I know selection acts on phenotype, I have phenotype to genotype map. Okay, so I'm at this level. That's the connecting thing, right? And I'm now in population genetics. I do whatever I want to do. Then work it out things and go back from genotype to phenotypic level. And then I know what's happening at the phenotypic level, right? So that's the sort of thing I was asking Uma to close the loop. Okay, but we know very little, unfortunately, about this map. And whatever little we know is pretty complicated. Okay, so that's why it's a difficult problem. Okay, so there are levels, at least you have to appreciate that, and there is some effort to connect them. So Archana is looking at some of those problems. Okay, so if this something excites you, it does to us, so please do speak to her. Okay, so let me start a little bit with Collison theory. So I will not prove anything, the math is really nice. So if there's something excites you, please do look at some of the papers. It's a book actually, yeah. Let's give you the name of the book by John Wakely. He also lectured here before, so you might want to look at his book on Collison theory. So what is this Collison theory? So let's go back, single locus and all that, okay, and Wright-Fisher process. Okay, so there are other versions also you can do. So I have these guys. At t equal to 0, that's the two of them. Or okay, 3. Okay, and I'm going to describe to you. Okay, we'll see. So I'm just evolving this population according to some rules, maybe Wright-Fisher. And I just made this genealogy. Let's say no mutations for now, and then I evolve it further. And so on. So this is the forward in time, right? Collison theory deals with genealogies. So suppose I'm interested in only 2 guys. That's my sample. So I have a large population of millions, okay? And then I just have a sample of size 50. So I'm drawing 5 things, but what I mean is a million things, okay? And I have a sample of size 50. So let's say these 2 are my samples. Okay? So Collison theory says you just ask who is your parent, who is your parent, and who is your parent. Like, you know, the way you will do your ancestry. So let's connect this. Okay, I think I didn't get the correct one. Okay, so I'll have to do it by hand. Okay, so let's say A1 and A2 are, what are they? Neutral. Okay, let's say they're all same for a second, okay, because I can't draw so much. They're all same types and neutral, okay, no mutations right now. So let's, this is our t equal to 0. Sorry about this. And this came from here. This also came from here. So, okay. So these two samples I had. You ask who's your parent? This is my parent. And who's yours? And yours. And this. Okay. So this guy was, what is it called? Common ancestor, most recent, I mean in this case it was the one because it just finished here. So this is called the most recent common ancestor of this sample, right? Okay, so this all of you know, very good. So what one is thinking of is making these kind of genealogical trees. But now I will not prove anything, I'll just define what's a Collison tree. What one does is not take things in discrete generations, 0, 1, 2, 3 generations, not like that. You have to do some math, and it turns out that if you if you run the time in some units, you basically, suppose I drew this guy, how does it look? It will look like this. These two samples, right? Okay. Suppose I had one more sample. How does it look? Like this. Okay. So this, going from there to this genealogical tree requires some work. That was done by Kingman. Okay. But this is called the Wright-Fisher process, and this is a Collison tree. So I know that you have seen this sort of thing, but what is interesting here is, what's the definition of it is the following. It's defined as the following process. That means if you take 3 such things, the time at which these 2 branches will coalesce. Everybody understand the meaning of coalesce? Joining, right? This time at which they coalesce is a random variable. Okay? And what Kingman showed was that this time, if I have 2 of them, let's call that time t3. Sorry, they have 3 samples right now. This is some time t3, which is a random variable. And then I'm now left with 2 of them, right? And this time is t2. So this ti is a random variable whose distribution is this. Is the definition clear? I choose 2. Let me explain once more. So if I have 3 samples, distinct lineages at this time, they will coalesce at a time t3, which is equal to, the average time of that is roughly equal to 1 by I choose 2, average time. Okay, so this time t3 is how much? I have 3 samples, no? I is 3. So I is the number of samples, number of genealogies. So how many I have here? 3. So time is, according to Kingman, 1 by 3 choose 2. Everybody knows what I mean? I choose 2. Oh, I should write it perhaps. I factorial I into I minus 1 by 2. So can you tell me now? 1 by 3. Okay. So when they have merged, then I have left with 2. So what is T2? Average T2? What happened? 1. Did I get it right? Yeah. Okay. So this is a definition of neutral coalescent tree. There are lots of things which has gone in. Okay. So but when you say coalescent tree, what one is asking is I have I samples and they will merge, these lineages will merge, but the merging time is a random variable, which is an exponentially distributed random variable. That is, this time scale is this, and the average time goes like 1 by I choose 2. That's the called coalescent tree. This is okay. This is a definition. I want you to do the following. So I have 3 samples again, or let's say 4 samples. The important thing here is that no 2 merges should happen at the same time. So if I have 4 samples, this is not allowed. That's the sort of thing which comes from maths. This is not allowed. Okay? So suppose I have 4 samples. Can you draw for me coalescent trees? I'll draw one for you and you tell me if you like it. So which one would you like to merge first? First one, second one, third one, fourth one. Which samples? Which 2 samples? Any of them? Huh? Second, third. Okay. Good. Okay. Which one next? Second, third to fourth. Right. So this is a coalescent tree of 4 samples. Is this the only one? Can you draw other ones for me? And is this a typical tree? What I mean by that is, the time here was 1 by 3, and this time should be 1. So therefore, this branch length should look longer, no? Right? Is this a good typical tree? So draw the geometries, that how these trees would look like. Is this the only possible tree with 4 samples? So draw which are the rest ones, and draw it correctly in the sense that it should capture this time scale. Achha, 3 is better to do first? 3 is better. Oh, sorry. So 4, if you're doing good, carry on. But if you're not doing 4, start with 3. If I have 3 samples, how many trees can you have? And draw it correctly. Okay? I don't know. Do all of them. Just do for 3 first. 3 done. All 3. Come to the board and draw. Sequence. No. Sequence matter means what? I mean, I think I'm looking for topologies, meaning the structures. Which ones come first? And then like 2, 3 can come first, and yeah, that's same. If I just say the labels, that's same tree. So let's just keep the same labeling and then try. Anybody wants to do? There's quite a lot of them, no? Let's do for 3 first. Come. No, so 1, 2, 3. 2 and 3 merge first, and then 1. Right? So it's a different sample. Then he's saying 1 and 2 merge first, and then 3. Right? Yeah. And yeah, thanks. And then 1, 2, 3. 1 and 3 first, and then 2. Different samples. Okay. So these are the so there's no unique coalescent tree. This was the sort of point I wanted to make. Now I'm going to make one tree, and I want you to tell me if it's correct. Is this it's possible, right? This tree is possible, why not? But is this a typical tree? Yes, they have to be, yeah. So this is the time T5. And this is T4. 1, 2, 3, 4, 5. Single merger. Is this tree possible, first of all? Yeah, why not, right? So that's fine. But is this typical? By typical, I mean average. Okay, sorry, angular brackets are. How should a typical tree look like? How does Collison trees typically look like? So I want you to answer that kind of question. Is this typical? So I'm not asking you to draw all the trees with 5 samples, but if I say the times are not Ti being random variable, but they are according to Ti average, how would you draw a 5 sample tree? A 5 sample tree. Is the question clear? Huh? I'm saying these times, so there are 2 sort of stochasticity, right? In the sense that you could merge first and second, first and third merge, and so on. That's one thing, right? Forget about that part. I'm asking about the time scale part. The time to merge is given by that expression, 1 by I choose 2. Average time. Okay? So, does that tree gel with this formula for average time? Yes or no? No? So how would it look? Let's say I coalesce in exactly the way I have done it. Could you please come and correct? What's wrong with it? Somebody said no. You are working coalescent trees, no? You will handle. Come, let's see it. How would it look? You have to draw it. You have to calculate that average time. Okay? And then use that information to correct that.
Once you increase the number
so I can just draw again. Don't erase mine. Okay? Is this correct? I think I have to explain.
So, if you increase the number of, if you increase the I here, the average time decreases. So, the initial coalescence will be pretty fast, and then the time increases, right? So, after the initial coalescence, as per this formula.
I've lost them. So let me repeat. Okay, let's forget 5. Why did he draw like this? Why did he draw like something like this? Nothing wrong with this. For the 3. Right? Is this okay with you? Is this a typical tree? For 3 samples? No? Why? Yeah, right. So just use that information. This time over here was 1 by 3, typical time. And later on it's higher time, right? That's why this is not a typical tree. Typical tree is like this, where this is 1 by, and this is 1. Right? Okay? So I'm asking the same question for, let's say, 4 samples or 5 samples. How would a typical tree look like? I just want a shape. I just want a simple character, qualitative character of it. How do you think it will look like? It's a tree. Tree has leaves. A tree has leaves. It has branches. So how do you think it will look like? Would it look like this? I'll draw something else. How is this time changing as we go back in the future or back in the past? The time is increasing. That's all I'm asking, guys. Nothing very deep. Right? So coalescent tree has this characteristic. The qualitative characteristic is that there are lots of leaves, tiny leaves. That means it looks something like tiny leaves here. And as you go back in the past, time keeps increasing the way he drew. These are shorter ones, okay? And these time scales keep increasing with time as you go back. That's a typical coalescent tree. It has that characteristic. Now, biologically, why do you think that makes sense or not? If it does, why? Or not, why? Does it make sense? So I have 10 samples. Coalescence occurred in some short time, let's say 1 unit. The second coalescent event took longer. third coalescent even, to decrease 10 to 9, it happened fast. To make 9 to 8, it took longer. 8 to 7 took even longer. Does it make biological sense? Or was Professor Kingman wrong? Right? You guys are throwing all these big words at me, phylogenetics and species and this and that.
Two scenarios. In an early birth scenario, basically when you have initial mutation rate was very high.
No mutation rate or anything, no. There's no mutation.
They are splitting based on some difference, right?
No, how are we splitting here? I'm just asking who is my, parent. That's all.
Okay. So even if that, why are we assuming that the initial organism took much longer to split than the later organism?
I'm saying this is what this theory gives, coalescent theory gives. And I'm asking you, does it make sense? Please take the mic. Yeah. Is this clear, first of all? How does a coalescent tree look like? Typical coalescent tree is, there are these leaves which are small, that means the mergers happen initially fast, and as time goes on, it takes longer and longer for the mergers to happen. Yeah.
I'm not sure about this, but if you have a well-mixed large sample from which you're picking your sample of individuals, then the probability that a few of them will have a common ancestor at a lower time is more. But if you look at more number of individuals, if it's a well-mixed sample, then obviously you would have a common ancestor much before, if it's a random enough. In that sense, this looks fine, right?
So when you say my sample, you're talking about the size of the sample or size of the population?
There is a population out of which I've taken a random sample.
Yes.
If that sample is random enough, then most probably it will look like this. A few of them will have an immediate ancestor, but a lot of them having a common ancestor immediately is very low. So they'll have it much.
So lots of them will have immediately, they will be able to find the ancestor more quickly. And then finally 3 will become 2. And difficult to find the ancestor, no? Right? And then 2 has to become 1 and they have to find each other. Right? So that kind of thing will increase the time. Does that make sense? Okay? Is it okay with all? We have an alternative explanation? Makes sense, right? That's what you have to say. I wouldn't know otherwise. Is it okay? Okay? So this is one characteristic of coalescent trees, which you should just remember because it's a simple thing. Okay? And it makes intuitive sense even if you don't remember the formula. Yeah.
So if we give numbers to that wrong tree, to the branches of that wrong tree from left to right, like 1, 2, 3, 4, 5. So those number in the correct tree will be like 1, 2, 4, 5, and 3, right? Or like
these numbers don't matter, no? That's, I mean, yeah. What is?
Yeah. So the same numbers in the correct tree. The other tree which he drew.
I mean, it's not, this tree is correct. There's nothing wrong with it. Because trees are random variables. But it's not a typical tree.
Okay.
That means I was asking you to make a tree. If I make all kinds of trees, I take these random variables and I just generate all trees. Lots of trees I will generate. Typical tree. Most of the trees will look more like this tree. That's all I was saying.
Okay.
This is okay. So what do we do with this coalescent tree business? Any idea? So we have made this tree, nice math and hoo-ha. What do we do with it? Is it of any use? So what do we find from it? But how would I find it? I don't know. I can't go back in time. All I have is these guys. I just have this information. This is what I have. So this is very static. This is not one experimental evolution or artificial process or something in which things are happening in time. I come tomorrow or day after and so on, look at my numerical experiment or my petri dish. It's not that. This is all I have. This is static. I got this 100 samples from human population for the world. What do I do with that? How do I know from that anything about the most recent common ancestor?
So, based on what samples I have, I can find this timeline using this formula.
How?
You I'm just substituting the value of I.
How? So keep going back. So how did that formula come?
So it's coming from the understanding that the time that it is taking is following the distribution.
Yeah, but how did that come? I haven't told and you haven't asked. Okay, so this is assuming these formulas are valid, assuming. So you start with an assumption that selection did not happen and the population size was very large and of constant size. So this is the neutral coalescent or the Kingman's coalescent. Okay, so I gave you the formula, but this is valid for assuming that there is no selection. Okay, you can pick mutation, that's not a big deal, that we can do, and population size is constant. So hold on. So I have this 5 samples, right, today. If I assume that today's sample is shaped by neutral evolution, right, so then these will be the tree, right, and therefore you would say this is the MRCA. But how do I test that it was neutral in the first place?
Your population size is
Meaning I'm doing this Wright-Fisher process in which, right, see, in million years ago or something, population might go through a bottleneck. So for example, it came from Africa or something, right? So but I'm assuming population is exactly constant. Millions remains millions all through, right? So assuming neutrality, I expect this tree and therefore I have some prediction for MRCA. But how would I know that it was neutral? Are you guys going to do it? You don't know? Oh, wow. How?
I understand why it is not neutral, would not. But the population size is constant. What is the problem?
Population is not constant. No, because I think you can sort of think of, no, so when population size is changing, you have to do slightly different thing, but roughly this goes through. Short answer.
You have, although when you have 5, you're connected.
No, this is the outcome from this. After doing something to it. But if this guy was 5, it became, after a few generations, 500, and it contracted back and so on. This Wright-Fisher process is very different. So the same kind of calculation you have to try and see if the population size is changing. So then the derivation, this black box, which I haven't told you about, that you have to look at. But generally it sort of goes through. You can cook up an effective population size and this basically goes through. Okay? Yeah. So what good is it? Okay, so I'll tell you one tiny thing. So what you can do is the following. Okay, let's do that. So let's have mutations also. And just look at 2 samples. So let's say I just have 2 samples and they merged in some time 1, right, according to that. But it's measured in some units. It's measured in units of n. These are all answers I'm giving you, okay? So it turns out that this merger happens in time n. Okay, that's what Kingman showed, okay, of the 2 of them. So now, if mutations happen, what's the diversity would you see? Mutations could happen on this branch per unit time. Mutations could happen on this branch per unit time with probability mu. Along this branch, how much time I had? N. So how much mutation is accumulated? N times mu. What about the other branch? Same. So 2n mu. That's the diversity you would see, denoted by theta, the standard notation. Okay, this is the nucleotide diversity according to, if I assume, so the logic is the following. You assume that population evolved neutrally and it has a mutation mu, then nucleotide diversity is given by the formula 2n mu. That's fine. But now the interesting thing is theta is something you can calculate from your sample. because that's all you have, right? So this you can find from your sample. All you have to do is to figure out how many differences are there. That's a diversity, right? Wherever they're different, you just count how many are there, how many pairs are different, and you just count that. That's all the diversity is. So this you can calculate from a sample, and that will give you an estimate of n mu. Few things to note here. First of all, what is it giving? It's giving me mutation rate? Is it giving me population size? What is it giving me? N mu. So this is the lesson which we learned yesterday also. Things come in this combination, ns, n mu, nm. Okay? So this composite parameter is what you can estimate if you assume that the population evolved neutrally. So I got this n mu. Still, problem remains. How do I know it's correct? This n mu is correct or not? Sorry? Yeah, tell. No, but we don't have enough fossils, right? I mean, there are lots of gaps. So without doing the fossil records and all that, how can we know? So again, I'll just tell you the answer. So people have calculated this object theta. It can be done in two ways. So, I mean, this one, since we didn't have time to go through in detail, I'll just quickly tell you the result here. So it turns out you can calculate theta in two ways. Something called theta W and theta, I forget what. Okay? Theta is a nucleotide diversity and number of segregating sites. Okay? From sample you can calculate. And the expectation is that they two should match. If they don't match, then it indicates that your starting assumption. So what's the logic? Assume neutrality. Sample I have, calculate segregating sites, calculate nucleotide diversity. Expectation is that they should be same. If they are not same, what's wrong? My assumption must be wrong. So people do very sophisticated tests like likelihood test, all kinds of things which I know very little about. But that's basically the idea here. So it's segregating sites are simply the ones which are not fixed. So if you look at some locus, and some are A's and some are T's and all, so that's a segregating site. But if other locus, everybody is A, everybody is T, that's a fixation has happened. That's not a segregating site. So I think Uma might do more of this. So maybe wait for her. Okay. So this is roughly the idea. So if first time when you hear about coalescent, that I'm just giving you this static sample, and you can still say something about what shaped its history. It looks very exciting, and I think it's a pretty exciting one. But it's probably a little too mathematical, or lots of simulations are required because it is a really difficult one. I'll just finish with one thing. If there is selection, then the coalescent tree is different. It's not a Kingman's coalescent. This is a discovery which was made a few years, like 10, 20 years ago. It has a different name. It's called Bolthausen-Snizman coalescent. Big names. Okay, I'll just write it for you, so you can savor the flavor of these names. This is the one with selection. Okay? And the other one is called Kingman's coalescent. Okay, fine. In the last 15-20 minutes, I want to give you one example of something from quantitative genetics. Okay? So you've done a lot of this, and I gave you a very quick flavor of this one, and I want to tell you something about this one now. Okay? Okay. So I want to introduce to you one classic quantitative genetics model. It's called an infinitesimal model. Okay, it's a short, like half-page thing. Okay, so don't worry too much. Okay, so I'll just take you through it. So this is another old one. Actually, I think in literature, you'll see lots of infinitesimal models. I mean, everybody has their own version of it. Okay, so I'll describe one of them. See, so the reason I wanted to do this to tell you how do we connect the genetic thingy to the phenotypic thingy, right? So because we said, we haven't done that, we have just been at the genetic level, what's happening at the phenotypic level, right? Okay, so I'll just introduce this very simple model. This is what, 1918 or something like that. See, the idea here is this. So how do you think people are able to say something about phenotype in 1918 when they knew nothing about genetics or knew very little about it, right? So, like for example, you heard about polygenic selection, right? So, in which you said that certain traits, they are determined or controlled by lots of loci, lots of genes which are under selection. So here the idea was that, let's say the trait, say height, it's controlled by a large number of loci, so large that are basically infinite. Okay? And the effect of each of these loci on the phenotype is tiny. So the effect A, okay, that's tiny. So if I have this height, okay, you have some height, you have some genotype, you have some height, okay, and if I make a mutation, if there's a little change there, it changes the height correspondingly. So suppose somebody has this genotype, and we call it sigma. So the height or the phenotypic trait Z, how do I find? I just told you this is genetics, this is phenotype, and you said I need the map. So it's a simple assumption you can make saying the map is additive. That is, if I have a 0, tell me one number. 0. If I have 1, I put an A and add them up. This is the additive phenotype genotype map. Okay, so that's the assumption I have made here. Okay, now again you will say, oh wow, the additive, hoo-ha. But you know, these are models are useful because they at least serve as null models. So like in neutral coalescent, if it's not neutral, that's the first thing to check. But there's nothing like neutral, is a selection or not. But if you don't match that, at least it tells you that it's not neutral, it acts as a null model. So you can think of the additive map the same way. Okay, so this is the height, if you add it up. Suppose I had a different sequence. What's the Z value? The height value of this guy? 4A. And so on, right? Okay, so this is my genotype, and that's my phenotype, and that's how I go between them. Okay, so now what I would like to do is to ask how phenotype is evolving and what does it say to me about the allele frequency. So I want to connect the two things. Okay, so let's do that. So let's say the frequency of zeros is P0 and frequencies of ones is P1. So going by that, can you tell me the average phenotype value in this population? So I'm thinking of a population which is infinitely large, okay, and there's some sort of selection which I'm not specifying. So what do you think would be the average value of Z bar? So these are the allele frequencies. How much would it be? So 0 into P0 plus A, P1 into L, because they're L loci. For each one you will put, no? P1 is the population average. Is this okay with everyone? Okay. So therefore, if adaptation is happening, if there is a change in the height, then dz bar by dt equals La dp1 by dt. Okay? Okay, that's fine. Now, who will tell me the variance in Z? So I think Vishnu was trying to do something like this. So you sort of know the answer already. So why don't you tell me what would be the variance in Z? Again, you see, we take things as 0 and 1. So by now you are experts. P into 1 minus P? P0 or what's my notation here? Is that all? Yeah? That's it? Huh? A square. Yeah. What is this? Some of you got this like quickly. What did these other guys do to get this formula very quickly? They just told us like this, na? What is this NPQ? Of binomially distributed random variables. Because again, Z is what? It's a sum of variables, right? Which are 0 or 1 and with a probability P0 or P1, right? So again, the same thing that you learned on first day. Again and again and again it will keep coming. So, please learn it properly. Okay. So, let's just calculate this because I want to get to a point. Can you tell me how much is this? So for some reason I'm doing the change in the mean, change in the variance, and I'll come to the point in a minute. Just tell me the answer, please. Derivatives, all of you are experts now. And let's wait for this. Let's wait for others. Because the sizes, because you have to think, we are adding up those numbers, their value not 1 and 0, it's A, 0 and A. So when you're squaring and all, would this expression have LA square or not? Yeah? Why? Because the constant, right? Derivative won't operate on the constant. Then derivative of P0 and P1. P1 is again 1 minus P0 also, right? So let's write it as P1 into 1 minus P1. These are the allele frequencies, right? So what's the derivative? P1 minus P1 square, so right? So is that what you got? Yeah, good. Okay. So the idea here is this. So one is thinking that this trait is composed of an infinite number of loci with the effect A is infinitesimal. That is, this is very large, lots of loci, but the effect is teeny, teeny, teeny. Okay? And somehow these work together, these tiny effects, they work together to make a change, a finite change in the phenotype. So dz by dt, this is finite. I mean, L is very large, A is very small, but this whole object is finite because phenotypic changes are finite, right? Phenotype changes. Okay? So this is some value, that's what I mean by finite, some value. But the classic assumption here is, however, the variance, the genetic variance in the population does not change much. So this is genetic variance. Where else have you seen genetic variance in these lectures in this week? Where does genetic variance come? Some words were being thrown now. Big words. Mutation? No. Where does it come? Yes. Heritability, right? The numerator has precisely this object, okay? And the denominator has phenotypic one, okay? Genetic variance is constant. Now, why and all, there are reasons for it. Part of the reason is that it's consistent with some data, and the reason is because for mathematical simplicity, okay? So, but that's what is generally assumed, okay? So, I need to satisfy these two conditions. L should be large, because why am I taking this L large and A small? Because I want to understand only the phenotype. I don't know what is happening underneath. I just know it's not a Mendelian trait. It's not just by one gene, right? So therefore, just one mutation occurs, the color changes green, yellow, and so on. But there's like lots of things, just I don't know, million of them, and teeny changes they're going to make. And that's all I'm going to say for the genetics, right? Because I don't know enough about it. Okay. So now what I want is, I want the change in the mean, this guy to be some value, but I don't want this to change. This change is about 0. Can you think of a way to do that? And I'm also assuming this. Can you square these three things? How this? So there's L, there's A, there's dp1 by dt. Right? So these are three variables that you have to play with. L should become very large, A should become tiny, and the dp by dt, you see whatever happens to it, such that phenotype changes, but not the variance much. So could you sort of come up with a suggestion? Sorry? Sorry? Yeah, so that's one possibility, that if they're equal frequencies, but that's a very special point, right? So we will not take that. And also it's saying that dp1 by dt is 0 then, because it's saying at half, right? So we will not take that one. So this is not 0. So it's going to 0. Okay, not exactly 0 right now. Can you come up with something? Anybody has done any quantitative genetics, even a teeny bit? Not at all. Many of you look at some morphologies of this and that, no? That's a phenotypic trait. So how do you understand that? That's quantitative genetics, no? As far as I know. Isn't it? So what do you guys do there? You must have encountered this, but maybe in a different way. No? Okay, I'm giving you 5 seconds. Come with suggestion. Suggestion I want. I just want a suggestion. No, I just want some value, a fixed value, constant in time. Some values. See, I have just 3 things. I'm just asking for suggestions so that these 3 things work. That's all I'm asking. And I'll give you one answer. Any suggestions? Hint, play with these guys. L is large. See, this very light is funny things, no? Infinity. There's nothing called infinity, right? Right? So what I mean is, L, if I keep making large and large, if I have this trait, which is between 100 loci, then the effect is some value, okay? Let's say 0.01. If I make it 1000, the value is decreased further, right? That's what we mean by taking these limits. Okay, I'll tell you the answer because time is running out. So, if you assume, and you should try it and see if you agree. If you assume that the effect size goes like 1 by root L, so root L is, okay, square root, everybody knows I think, right? So that if L becomes large, A becomes small, but in this specific way, okay? If I assume this, what happens? This means A square goes like 1 by L, which means L A square is constant, say 1. So then what will happen? This is a constant, and therefore allele frequencies must not change, not much with time. So dp1 by dt is approximately 0. Why is that okay here? This should be finite, right? This L A, and if I assume it should go to 0 like, how should it go? No. Okay, I think I'll just give the answer. It's okay. So, if I assume that allele frequencies go to 0 as 1 by root L, okay? You get it right? Yeah. If the change in the allele frequencies goes like 1 by root L, size goes like 1 by root L, then all these 2 requirements work out. What does it mean? What it's saying is that a quantitative trait is controlled by lots of genes, each of a tiny effect, such that the genetic variance does not change, but the phenotypic trait value changes, and allele frequencies don't change much. So allele frequencies are going like this. Starts with some value. That's what this is saying. You can go through it, it's simple math, but let's just skip that. But that's the upshot. And I want to leave with the simple question. Is this a kind of typical picture have you been seeing in all these lectures? The quantitative genetics picture is that there's a trait with lots of genes and the adaptation occurs such that allele frequencies change little. Is this what you have been seeing? What did you see? Is that what you saw in many of times we've been drawing allele frequencies? Starting from? Yeah, and yeah, 2 to nearly 1. Wow, yeah. So, this is not what we've been seeing, no, all this time. We have been talking about things like this. So, what's going on? Was yesterday's whole thing was wrong? Or today's this last half an hour was wrong? Or both are okay? Or what is the answer? So, this is what I said in the first lecture. These are called subtle shifts or small shifts in the allele frequency. This is a typical quantitative genetics picture, and these are called sweeps. And this is what you get from typical population genetic picture. So we thought we were trying to connect the two levels. But look, this guy says if I just stay in the universe of population genetics, there should be large changes. Things go from 0 to 1, big changes, right? Okay, somebody might mention hitchhiking yesterday. Okay, but if I am looking at some morphological traits or some phenotypic trait, there are tiny changes in the allele frequency. So, what do you think is going on? Is something going on? But what is correct? No. I can look at allele frequencies of that, Galapagos finches or something. This picture is still for single trait. No, what is it called? Pleiotropy or something.
No, we were only looking at one locus, right?
Oh, no. Now, even if you do, lots of loci. Morning what I did, adaptation, asexual adaptation, I said frequency is going. Yeah, so that is still true. So it's not about loci. So there's so many theories and so many answers, and both are seen. So it's not like theoretical something. Both are seen, small changes as well as large changes. So both are there, but one set of theory gives this, another set of theory gives that. So, both are there, that's what I said. Both are there, people see that, both of it. But No, this is parameterization, no. Yeah. No, so if I do this properly, so what you're saying partly is right, yeah, okay, that's one. But I am thinking of these polygenic traits. Even if I do only population genetics, with lots of loci, okay, then also I'll get sweeps. I'll get big changes. That's what I described in the morning. It was not a single gene, no? I said, I'm thinking of a very long genome. Okay, so I think this is a very short break or tea break? Tea break, oh, this is a short break, right? Okay, so I think we should stop. Okay, so I'll just leave you with this question. So what is this big tension? So first of all, we don't talk PopGen guys with the QuantGen guys. QuantGen guys don't talk to PopGen guys. And when we talk, we find we don't agree. So, I don't know. Okay, I'll stop.